So I will continue with this aromatase inhibition example, but probably try to make it a little bit more illustrative and understandable. Hopefully you already understand it by now, but let's do some cartoon animation. So we have, if we have testosterone actually converted in the fish by aromatase to estrogen, this is the normal egg production process, by the way. So this estrogen actually goes into the liver and provides the liver ability to generate vitellogenin, which is incorporated into bloodstream. From the bloodstream, it goes to the ovary and actually facilitates the normal egg production. What happens if we have a chemical like fadrozole, which interacts with the aromatase inhibition process? So basically, it slows down the conversion. We have less estrogen. Then this estrogen doesn't activate the vitellogenin production, and we have less fish. Unfortunately, that's not part of the pathway. It's just for keeping you awake, I guess. So what if we want to try to describe this process, a stepwise process, and basically encode all of the elements as a pathway? So the first step is fadrozole interacting with aromatase, and we have the chemical drawn here and the fadrozole as a molecular initiating event. Then we have reduced E2 synthesis. Then we have the reduced E2 concentration in the liver, less vitellogenin produced, again, reduced circulating vitellogenin, reduced uptake, decreased spawning, and population reduction. So basically, this is how we define our pathway based on the really important key events that happen on the way. And maybe here I would like to make a little bit of a deviation. On the subject again on linearity of the pathways. So if we look at this, this is really nice and linear up to the canon. But the idea is basically if we have processes that are maybe feedback loops and things that actually accelerate certain processes or lead to different branches that actually don't produce population reductions. Those actually could be incorporated in the links between the different steps, and we don't need to show them necessarily as the branches of the pathway. I wish actually show you more examples in the Effectopeida itself, where we have branched pathways, and some of those branches are actually biomarkers that could be used for enforcing your knowledge. The main idea is when you look at all of those complex things that happen in biology, just pick the ones that are really the most essential and drive the understanding of causality of the processes. So if simultaneously multiple things happen, we can definitely mention them as a separate branches, but if we want to outline which one we think is really the most important one, we just mention this one. If we have cases where two things really need to happen, so we will just draw them as two things. So it's conceptually, it's not a limitation, it's just a preference. So again, sorry, if you look at the pathway that we just drawn, we can actually think of what is the taxonomic relevance of this pathway, and we can see that the first three steps are actually things that are preserved across amphibians and vertebrates, and the following steps are actually valid most likely for egg-laying creatures. So basically, with defining this pathway for fish, we already have some kind of knowledge what will happen in other species, maybe at different rates and with a lot of other modifying factors, but still we have the initial idea. So what this diagram of the AOP doesn't show up when we just draw it like this, this is just a sequence of events, and we just miss all the biological context that I was showing in the previous slides. And if we want to, again, acknowledge the taxonomic relevance, sex, life stage, level of biological organization, generations and so forth, we actually need to introduce something to be able to show it and encode it in the pathway structure. So what Effectopedia does is basically provides organizational space, which is called pathway space, and it has those multiple dimensions describing different aspects of biological context. So we have, if we look at the 2D projection of this multidimensional space, and here the same example that I showed just as a linear chain is shown in this 2D projection. So we have fadrozole, again, interacting with aromatase. And you can see that this is drawn in a kind of a cell where we have the vertical axis is actually sex, and the horizontal axis is a level of biological organization. So everything that happens on molecular level for both male and female is going to be located here in Effectopedia. And here, just for illustration, is given another one of the other biological contexts that are applicable. So this is basically amphibious and vertebrates, where the whole first few steps of the pathway are valid. But in actual interface in Effectopedia, you can see the full list of other biological context dimensions. So if we look at this pathway a little bit further down, you can see that on organ level, things are happening only for female. So yeah, it's kind of obvious because it's laying the eggs, but also you can see that the adverse outcome, because it's defined on population level, it's also for both sexes. So the idea in Effectopedia is that you actually can switch between those different dimensions very easily just by clicking on the axis and choosing another one. And also the list of those possible dimensions are things that you can define by basically user definable, and also could be really something that's relevant to your pathway. So in our experience, for example, we did look at one of the pathways that has a multiple generation study of medaka, and we needed to add an axis for generation just to be able to display the pathway in an appropriate manner, and we can have a split between the pathway, something that happens only for the first generation and another thing that happens for the second generation, and so forth. Also, I mean, I would like to take a brief moment to basically look at what are the elements that are building the pathway itself. So if we again zoom in to the first few steps of the pathway, you can see the chemical, and chemical comes with its usual like cheminformatic description in Effectopedia, and also is something that's basically more or less structured like a Euclid. So it has two levels: a physical substance, which could have different impurities and so forth, so we know exactly what could be tested in general, and another layer which is just the structure and so forth. Physical substance is actually more applicable when we go to the test methods. The thing that actually links the chemical with aromatase inhibition, in more general terms in Effectopedia, is called link, because we have link basically working between chemicals and outside of the AOP framework. So this first step is not key event relationship, is just a link. And this link actually has some form of explanation why we think, in this case, fadrozole interacts and how interacts with the aromatase. And we also could provide the dose response data, which support this interaction and show in a real assays. So when we have look at the link that's actually between two key events, it has similar structure. So it has a quantitative part of the description, which provides all the biological plausibility of why we think those two key events are related, and also has the quantitative description, which could be quite different compared to the one that I just shown in the chemical case. So we can have direct relationship, which is basically whenever the first key event happened, the second one following the upstream, the downstream effect actually always happen in the same proportion. We can have proportional, we can have threshold relationships, and in the most like universal case, we can have response curves in which we basically map how much of the input, let's say key event, which is here, causes how much of the output. So when we describe in Effectopedia pathway, basically those elements that I currently shown describe the topology of the pathway, but the evidences themselves, so supporting evidences, are separated in their own visual primitive. So we have different types of tests that are represented with, I mean, they are color coded depending on if we have in vitro, in vivo, in silico, and so forth. And they also use the same pathway space, so they have location in the same way the boxes of the pathway key events have. So if I want to show, for example, a few of those test methods, those are basically connected with a transformation function to the molecular initiating event in this case. And what you can see is that those are the first test example. Those are human cells. That this is a cell line of cancerogenic cells, human cell line. And yeah, the last time I checked, humans don't lay eggs, so we really need to make this assay actually report something that's relevant to fish or egg laying animals. And that's why we need this transformation function in general. So we have here something that happens in vivo, and here we have something that happens in vitro. So the toxicokinetic information might be part of this transformation function, also a lot of other considerations. So a lot of things are currently hidden in this chart in this simple f of x, but it's quite tricky to define in general. But if we have this, we also have, alongside this transformation function, we have the uncertainty of this transformation encoded. So we exactly, we will be able to know and justify how much we can trust certain assay to predict certain key event or molecular initiating event in this case. So hopefully we will be able to basically have a plain field of all the available assays for a certain key event and know in which case for specific chemical which one will provide the most accurate and predictable answer and reproducible and so forth. So there are a lot of things that go beyond those boxes as details. So again, if we look at what other assays are currently associated with this example, there are some TOX21 assays, and the last one is actually in silico model, which is basically a QSR model that uses the structure of the chemical to bin the possible potency of the chemical as a aromatase inhibitor. And again, here this is just an illustration of how the framework could integrate various different evidences and basically show them in the same framework, so you can compare the different methods and can see how well they are managing to compete against each other. And one thing that also could be mentioned is that basically all of those test methods are things that could be described as isotap test methods. So, but I just maybe want to say too much in a very short description. But basically, the test method itself has a description of all the measurements that are made, the conditions and everything else. So this is really relevant when you define the transformation function. You really need to know how much of the real biology is modeled in your test system, so you can know how much needs to be transformed to the in vivo case. So again, at the end I will show and let you know where you can find a little bit more information. So when we actually look at this pathway again, we kind of already know that in biology there are multiple ways to cause reduced E2 concentration. And in this case, just a few to mention is like estrogen receptor agonism, androgen receptor agonism, and CYP inhibition, and so forth. So again, like in the wiki, we have to define those key events that are common between pathways just once. And additional simplification in Effectopedia is when you actually define a new key event, the system always looks at the same cell in the pathway space what's already available. So it brings a list of existing key events that are more or less in the same biological context. And if you see one that's relevant, not only by title, but you can see a preview of description, then you can say, okay, I mean, I can use this one and grab it for the pathway. And the advantage of this is also when we think about defining those quantitative relationships between these key events, we really need to define them once. And once we do that, if we can attach another MIE at the beginning of the pathway, we have a lot less work to do next time we want to do quantitative predictions. So the merit of basically accumulating those different and separate pathways in a pathway network is really very big, and that's why from the very start Effectopedia was designed to handle and display multiple pathways at the same time, and also use them potentially in the very near future at the same time, so you can choose and look what happens in all the available knowledge in Effectopedia. So one example of how this could be used is if we have a chemical 2, for example, and we have a slider for its concentration, and we start increasing the concentration, you can see that the first AUP number 3 activates, then at higher concentrations AUP number 2 activates, and very high concentrations AUP 1 activates. And the interesting part of this story is that we can look at this as if those 3 pathways were different organs in a single organism, you can say, well, MIE 3 up to outcome, adverse outcome 3, this is the most vulnerable organ of this particular individual species. But if we look at this across species, for example, if the pathways were defined for different species, we again can do the same, and we can say of all species that are encoded in represented in Effectopedia, this is the one which is most vulnerable to this adverse outcome. So basically there are a lot of things that could be done with the networks that a single AOP wouldn't really inform us enough. Then if we look at another case, we have chemical 4 activating pathway 4, but not completely, and we have additional chemical exposing the same, let's say, animal, and we see that basically the activation of key event of the second pathway influences the rate or quantitative transformation between key event X and key event Y, which actually produces the full complete effect for the first pathway. So the idea is that we can have those interacting pathways, and the pathways can interact in basically two major ways. One is as modifying factors like that, so they modify the rate in which certain process occurs, and another is basically directly having branches from one pathway accumulating the effect and influencing another pathway. So framework will allow both. We just will see how it progresses with the filling up the information. And finally, when we have multiple pathways defined and we have shared key events, we can look for those key events as things that are most likely to be better to be basically tested with alternative methods, since those alternative methods will produce a lot more effect, a lot actually easier maybe, and less resource involving results. So those are basically the priorities if we choose to select what really needs to be replaced with non-animal tests. Then those will be the ones that are really likely good candidates. And yeah, as a overview and summary of what I try to really squeeze is that Effectopedia really tries to do a visual representation of the semantic information that goes to describe an AOP. And what I didn't show here is when you click on any of those boxes, you get a lot more dialogues with the appropriate description of the elements. Then everything that's put in Effectopedia is version controlled, so you can look at older versions. You can cite your own version in your scientific publication, and this will be always the version that people will get at when they click on the link. And the advantage of this is that actually this doesn't really prevents the pathway from evolving, so it could evolve further down and have multiple other authors by the time when somebody clicks on the link. But at the same time, it's very easy to see and like to provide credit for the authors of the pathway, so everybody is really easy identifiable who did the contribution and what was done. Also, the review process, similar to the wiki, allows the pathway to be reviewed as a certain revision and again decoupled from the evolvement of the pathway itself. So if you want to take a look at Effectopedia itself, it's open source and free software. You can go to SourceForge to download a version of it. And also, if you look, for example, in YouTube for Effectopedia, you will be able to see a webinar which has a lot more details about the system. Currently, we are working on 3 examples which describe quantitative pathways. Some of them has a lot more information about the toxicokinetics and things that are actually prior to the AOP itself. But again, this is not maybe very easily describable in a few short sentences, but hopefully with those examples we would capture quite a diversity of possible uses of Effectopedia. Some of the examples will contain converted MATLAB or R models that could be computationally executed from Effectopedia and the results directly used, so you can see the predictions, what those models give, and you can also see all the assumptions that are made behind the model, so about the biological constants that drive the process, also the chemical specificity of the models, and so forth. So hopefully this gives some appetizing information to look for more details. And with that, I guess we can have questions for all 3 of us. Yeah, definitely. Actually, if you have, basically, you could have a lot of evidences that are contradictory as well. And the main driver behind providing so many details about the assays, the way the chemical was tested and so forth, actually hopefully will help resolve those conflicts. So if, for example, a chemical wasn't tested above certain, let's say, solubility level or something like that, you will be able to pinpoint why some tests give positive results, some other give negative results. So by just digging into the details or referring to those details, I guess experts will be able to judge for themselves which evidence from conflicting case is working for them. And also there was another very specific requirement that I currently heard was that when you use basically a pathway more for a predictive mode, then certain agencies might have certain preferences of what assays they want to use. And this also should be very much customizable in Effectopedia. So I guess US agencies will use one set of assays, OECD-based countries unfortunately will use slightly different set of assays, but that's the reality. Unless everything becomes harmonized, Effectopedia will be really open to adopt as many forms of legislations as they exist. So, actually, no. Effectopedia was the originally system that was open from the very start. So it's currently open as well, and it has 17 pathway sketches, some more advanced, some less advanced, in the same way the wiki principle works. So it's really targeted collaboration as well, especially because we tend to think that not a single group will be able to cover the whole AOP, so it's most likely actually group to contribute parts and fragments of AOPs instead of whole AOPs, and that's what we are hoping to encourage. And there are quite a few strategies that I currently am trying to think of to actually make Effectopedia populated. One of them is maybe something that we currently are talking about, like prizes to create incentive for young scientists to start populating this information from classical textbooks to Effectopedia and the wiki. So converting the existing more or less textual descriptions of information to something more quantitative and more computational. What I didn't show is that a lot of the fields in Effectopedia are going to be, and some are already, but some in the future will be basically semantically encoded and ontology based. So you can do a formal reasoning with the information that's entered in Effectopedia, and you can detect conflicts of information and a lot more elaborate things that will come out of it. But again, that's a little bit further down on the road because it's a lot of things needs to be done. And yeah, also another strategy for populating is maybe when I see so many, for example, alternative test method developers here in the SOT, I will be inclined to invite them to contribute to Effectopedia describing their own assays and maybe figure out one or two key events to which they are applicable and usable. If they describe their transformation function as well, that will be even bigger bonus. So we will have those basically fragments of information in Effectopedia accessible for others basically directly to reutilize and use. So, and also a lot of the information doesn't need to be entered. We currently use MBIT as a chemical and experimental data storage, let's say, server organization. So it will have ToxCast and all of the other, a lot of other publicly available data already imported in Effectopedia without you need to re-enter any of this information. So that's all about it, I guess.